All right, everybody. So this is going to be our e-learning version of the start to cell energetics and really the start of the molecular genetics unit in biology. So again, we're looking at molecular genetics. And let's take a moment and just go over the objectives of this lesson, of what you should know when this lesson is over. Just a quick reminder, you should be taking notes in your written notebooks, of course. Have all of your other devices away, just like if you were sitting in front of me in class. All right, so what we're learning today, we're going to first of all review the nucleotide structure. For my e-learners especially, you did not get as much time to sit down and really map this out with Play-Doh like we did in class, so I want to make sure I go through this with you step by step today. We're going to review the overall structure of DNA. Any upcoming quizzes or tests, you want to be ready to be able to draw the nucleotide and draw the full DNA structure. When we draw DNA, we're going to draw it with at least 4 rungs of DNA. We're going to be looking at photosynthesis and cell respiration, kind of taking it to the next level, looking at the specific formulas for each. We're going to talk about how the products of photosynthesis will become the reactants of cell respiration. We'll take time to review what a chemical reaction is, what do we mean by reactants, and what do we mean by products. And then lastly, we're going to take a look at different factors that are going to impact the rate of photosynthesis. So make sure you have these lesson objectives down, and then we'll start into our nucleotide structure. All right, so first of all, we're going to take a look at the structure of a nucleotide, but I want to review and make sure that you understand that when we talk about a nucleotide, we're talking about the building block of a nucleic acid. We're talking about the building block of DNA. Okay, so each DNA molecule is going to have 2 sides to it, like the sides of a ladder, and those sides are going to be what we call the sugar phosphate backbone. So as you can see here on the left, I have one side of the ladder, or one side of the DNA, and on the right side, I have the other side of the ladder, or the other side of the DNA. In those sides of the DNA, we're going to have a sugar and a phosphate, and then on the outside of those sides is where the rungs of the DNA come into play. All right. Next, let's take a look at the specific structure of the nucleotide as a review. So as I zoom in here, pause at any time so that you can get this drawing down, but I'm zooming into one side of the ladder. When I say ladder, I'm referring to a complete DNA molecule. So on that one side of the ladder, as we move from the outside in, okay, we're going to first of all encounter a phosphate group. So that's what this is right here in blue. This is our phosphate, okay. That phosphate is going to be connected to a ribose sugar. It's important to make sure, guys, that you're putting the oxygen in the correct location at the top of this ribose sugar. And then we have our nitrogenous base. Okay, so this part, I'm going to use yellow to show it, but this part right here now, this is going to be the start of the rung of the DNA ladder. Okay, so we've got our nitrogenous base there. I want to ask you a question here. We'll go back to our water section. In water, we learned that there was a specific bond that was between the individual molecules in water, the individual atoms in water. So if I had my water molecule, I had my oxygen, and then I had 2 hydrogens that were connected. What was the bond, though, that connected those atoms within a respective water molecule? Okay, so we should have that those bonds in the water molecule were covalent, and it's going to be the same in a nucleotide. These bonds right here, the bond that's first connecting this phosphate to this ribose, those are going to be covalent bonds. Okay, those are both covalent bonds. And then the bond that connects the ribose to the nitrogenous base is also a covalent bond. So this is one nucleotide zoomed in now to the side of the DNA molecule. Next, let's go ahead and draw this graphic organizer in our written notebooks. We're going to identify the DNA bases, and we've already talked about what the DNA bases are for the respective groups for DNA, but this is going to break it down even more to a chemical level. So we have two groups of DNA. We have what we call pyrimidines, all right, and we have what are called purines, and this is how we group the DNA base pairs. Pyrimidines are made of cytosine and thymine. We denote cytosine with the letter C and thymine obviously with the letter T, and then guanine and adenine, and we delineate it there with G and A. So make sure you've got this down in your notebooks, and then I want to ask you another question similar to the water question. So when we're looking at water, we said we had our individual water molecules, and they were bonded via polar covalent bonding in terms of within the molecule, but then there's also a powerful bond between those separate molecules. What was that bond called? All right, so that would be the hydrogen bond. So it's going to be the same thing in DNA. We're going to have hydrogen bonds between these. So we have to know how many hydrogen bonds there are. So between cytosine and guanine, there are 3. And that's simply because there are 3 locations for hydrogen bonds to form. And then between thymine and adenine, there's 2. That's because there are 2 locations for hydrogen bonds to form. All right, so on a quiz, for example, you might have a question that would say to draw a DNA molecule with at least 4 rungs of DNA, okay, and that's what we're looking at here. Is a DNA molecule with 4 rungs. All right, so this would be the type of question that I would be asking you. So this would be a good time just to pause the video and go ahead and get these 4 rungs down. When you're drawing these, you would want to label you just really need to label one of the nucleotides. You see how I have the phosphate, the covalent bonds, and the ribose labeled. You want to denote, make sure that you've got the locations of the oxygens. Make sure you've got those labeled. Okay, so on this side, my oxygens are pointing up. On this side, my oxygens are pointing down. This is because DNA is anti-parallel, and we will be getting into that coming up. And then also make sure you label your hydrogen bonds as well. So go ahead and pause the video, take some time, get a nice drawing of this DNA down. Notice the base pairing rules, okay? And then we're going to move on to the photosynthesis and cell respiration portion of today's lesson. All right, moving on, let's do a quick review of what a chemical reaction looks like. So on the left-hand side of a chemical reaction, we have what are called the reactants. These are just like the ingredients that you would put into a recipe before you put it into the oven. And then the products are the final product that you pull out of the oven. All right, but with photosynthesis, we're going to have reactants like light and water, and then products coming out like oxygen and, of course, glucose, the very important molecule of glucose. So let's go ahead and dive in now to the specific chemical formulas for photosynthesis and for cell respiration. All right, for photosynthesis, on the left-hand side, we've got, again, the reactants. So we've got photons of light, and make sure you have this down clearly and completely in your notebooks. Photons of light and water and some carbon dioxide. I think we all understand from middle school that plants take in carbon dioxide, they give off oxygen. We've got our photosynthesis reaction there in the middle. This reaction is happening in the chloroplast, and we're producing our molecules of glucose, some oxygen, and then there's always going to be a little bit of water as a byproduct in all of these processes. Take a moment and brainstorm, what is the organelle in the cell, in the plant cell, that is responsible for holding all of this excess water that's going to be produced and kind of held around during the process of photosynthesis? Our next reaction is going to be cell respiration. So we're taking, again, one of our big ideas today is that the products of one process become the reactants of the next. So we've got glucose here, which is a product of photosynthesis. We've got oxygen, which is a product of photosynthesis. Going through cell respiration in the mitochondria, the powerhouse of the cell, producing some ATP, producing some carbon dioxide, and producing some water as well. Okay, there's going to be three different examples that we're going to look at here, but specifically we're going to talk about anaerobic cell respiration, aerobic cell respiration, and the process of glycolysis being the third part of cell respiration we're going to dive into. Let's do a little comparison, a little T-chart here to compare anaerobic to aerobic cell respiration. Anaerobic means it's going to be without oxygen or in the absence of oxygen. Aerobic respiration is going to be in the presence of oxygen, or just really a higher concentration of available oxygen than anaerobic cell respiration. On the anaerobic side, in animals, examples, if we take track and field as an example, the sprint events, the 100 meters dash, the 200 meter run, the 4x1 shuttle run, those types of things are going to be anaerobic, meaning that the muscle tissue is going to have a little bit less of a presence at that time. The aerobic side is going to be our long distance running, the mile run, things like that. Now, that doesn't mean that if you're doing a long distance run that you're not going to have some anaerobic environment, maybe if you're sprinting at the beginning or the end, but by and large, those are considered aerobic activities. Taking a look at the plants, the plant example, so in anaerobic respiration, we're going to call that fermentation, and in aerobic, it's going to be just the regular life cycle of a plant, okay? So the products in anaerobic cell respiration are going to be our alcoholic fermentation or alcohol there, and then in animals, it's going to be what we call lactic acid. That's what causes the burning sensation in the muscle tissue. Anaerobic respiration is going to produce approximately 2 ATP, and aerobic respiration is going to produce about 32 ATP. All right, in glycolysis, we're going to be looking at both aerobic and anaerobic respiration. It's going to start in the cytoplasm, and glycolysis is basically taking glucose and breaking it down. So we're going to call it glucose breakdown. And we're going to be breaking down glucose. This is going to be what's going to help to produce that energy, that adenosine triphosphate or ATP that we've been talking so much about. Go ahead and take a moment now and go ahead and get this down on the process of glycolysis and the breakdown of glucose. So we're taking glucose, C6H12O6. We're going to break it down into 2 3-carbon molecules. Each one of those molecules is called a pyruvate. And that is then in turn going to produce our net of 2 ATP. The next topic we're going to take a look at are going to be the factors that are going to impact the rate of photosynthesis. And there are 3. So go ahead and answer here in the Ed puzzle. What do you think those 3 processes are? 3 things that impact the rate of photosynthesis. All right, so light intensity, temperature, and carbon dioxide concentration are going to be the main factors that impact how fast or slow photosynthesis takes place. All right, so for the first one, it's going to be temperature, so make sure you get this down. In this graph, you'll see on the x-axis, we've got temperature. And then on the y-axis, it's going to be the rate of the reaction, so the rate of photosynthesis in this case. So as you can see, as the temperature increases, as it gets warmer and warmer, the rate of photosynthesis increases. So a good example of this would be like growing tomatoes in Florida. This time of year right now, it's almost the perfect temperature. So on this curve, we'd be probably up here towards the top, right, kind of right around here. And then come the summer months, the temperature is actually going to be too high. So as it says here, I've got that above a certain temperature, the actual enzymes that drive photosynthesis, they're going to denature, they're going to break down, and the plant growth then will stop, okay? And that will cause this decline. to go down. Okay, so plant growth at that point completely stops because of denaturation. Make sure that you review the definition of denaturation from your enzyme notes. The next factor that impacts photosynthesis, or the rate of photosynthesis, is going to be how intense the light is. All right, so plants only have so much chlorophyll, right? They only have so much of this pigment, or the solar panels, as I like to call them, for a plant. So once the chlorophyll has absorbed as much light as it can, we're going to see this plateau right here. Okay. Another garden example of that would be, for example, I've got sunflowers all around the house. I have some that are in areas that get about full sun, 8 to 9 hours of sun a day, and some that just get 6 or 5 hours of sun. So those sunflowers aren't as tall. The ones that are maxing out here at the top, though, they grow the tallest, obviously. So again, chlorophyll can only absorb so much light. So as the rate of photosynthesis increases, it's increasing with light intensity. As the amount of light intensity increases, so does the rate of photosynthesis. All right, and then we get that plateau. It's going to be very similar as we take a look at carbon dioxide concentration. The graph is going to be very similar to this one. All right, so for carbon dioxide concentration, there's going to be a certain amount of carbon dioxide that's needed. There's going to be an optimal amount. That's kind of what we're seeing up here at the top. But this is going to be our general trend. As carbon dioxide concentration kind of increases, the reaction rate is going to increase until it gets to its optimal amount. And that's because there are enzymes that are responsible for what we call carbon fixation. Once those enzymes for carbon fixation are 100% saturated, then the reaction rate will level off, and that's where we get our plateau part there.